Okay, so welcome to this next video in which we are discussing the effect of sympathetic nervous system stimulation on the heart. Okay, so we have seen how the sympathetic nerves, which are going to the heart, they synapse on the cardiomyocytes of the entire heart and release noradrenaline onto it. And this noradrenaline causes all the cardiomyocytes of the heart to beat with a greater contractile force, i.e. To have a greater inotropy, which just means force of contraction. Okay, and this means that the fraction of the blood in every single one of the chambers of the heart that is ejected when the walls of that chamber contract is going to be greater because we're putting a greater force into that contraction. Okay, now what we want to look at is the other aspect of sympathetic stimulation on the heart. We want to look at the positive chronotropic effect, which is the increase in the heart rate that the sympathetic stimulation causes. So we want to look at the positive chronotropic effect now. Okay, and it exerts this by acting on the sinoatrial node of the heart and increasing the rate at which the sinoatrial node fires. So the sinoatrial node is the pacemaker of the heart, and it's made up of a bunch of non-contractile and instead conducting cardiomyocytes. Okay, and these cardiomyocytes, they produce spontaneous action potentials at a rate of around 70 per minute. Okay, so what we're going to do in this video is we're going to start off by just having a quick reminder of the way in which these sinoatrial node cells produce a spontaneous action potential at around 70 beats per minute. I.e. we're going to have a reminder of the SA node action potential, and then we're going to discuss how beta-1 stimulation by noradrenaline causes the sinoatrial node to increase the rate at which it generates action potential. So at rest, it's generating them at around 70 beats per minute. Okay, now the way in which the sinoatrial node cardiomyocyte produces a spontaneous action potential is by having a channel known as an HCN channel in its membrane. Specifically, the HCN channels that are most prevalent in the heart are the HCN4 channel. Okay, right, so HCN, by the way, stands for hyperpolarization activated and cyclic nucleotide gated cation channel. So the hyperpolarization activated and cyclic nucleotide gated cation channel. Hyperpolarization activated and cyclic nucleotide gated cation channel. Okay, right, so let me discuss the structure of this HCN channel with you. Cation channel. Okay, so if I draw it larger down here. Okay, right, so here is the HCN channel zoomed in on. Okay, so it's a nice big channel here. Right, so it's a tetramer. Okay, so it's made up of 4 separate proteins. So 1, 2, 3, 4. There we go. So this is the HCN channel here, and it's a tetramer of HCN proteins. Okay, and in the human genome, there are 4 genes encoding for HCN proteins. So there is the HCN1 gene. Then there is the HCN2 gene, the hyperpolarization cyclic nucleotide gated 2 protein. Then there is the HCN3 gene, and finally the HCN4 gene. So there are 4 genes all encoding for proteins which can be used to make a quarter of the HCN channel. So each one of these proteins makes up just one of these subunits that tetramizes together to make the HCN channel. So I've drawn this in blue. Here is one of these subunits that makes up the HCN channel. So, in order to make an HCN channel, you have to put 4 HCN proteins together. Okay? Now, the question now occurs to us, do you have to put 4 of the same HCN proteins together to make a tetramer? That would be a homotetramer if you did that. And the answer appears to be no, basically. You can make HCN channels which have different HCN proteins in each of the 4 sockets, a so-called heterotetramer, hetero meaning different. Okay, right, so which HCN proteins then are most expressed in the sinoatrial node cardiomyocytes? Well, basically it is this HCN4 protein here. So HCN4, the hyperpolarization activated and cyclic nucleotide gated cation channel protein 4, it's the most expressed in cardiomyocytes. So you have huge amounts of this HCN4 protein. You also have little amounts of HCN1 and HCN2 as well, but nowhere near as greater concentrations of HCN1 and 2 as you do HCN4. So you make absolutely loads of HCN4. So, what sort of HCN channels then do you think you are going to make? If you've got loads of this and very little of this, then most of the HCN channels you actually make are going to be homotetramers of HCN4. Or if they're heterotetramers, then maybe just one of the proteins will be a different HCN protein. So most of your HCN channels are going to be homotetramers of HCN4. You may well get a few homotetramers of HCN1 and HCN2, but mainly they're going to be homotetramers of HCN4 or a few heterotetramers in which the main component is HCN4. Right, okay, so that's what an HCN channel is, but it doesn't really tell us why they're so important, and they are so important. Now, they're so important because they have very different properties to normal voltage-gated channels. And the key thing is this word here, this hyperpolarization activated. Okay? So these channels are activated to open when the electrical potential difference across the cell membrane becomes hyperpolarized, not depolarized. The usual scenario is that channels become activated when they're depolarized, but these become activated when they're hyperpolarized. So, when the electrical potential difference across this membrane is negative 70 millivolts, okay, which remember means, so it starts off at negative 70 millivolts. Well, this is where we'll start the story from, although it's a cycle, so there is no beginning, but we have to pick somewhere to begin it from, and this is the most logical place to begin it from. So, we're going to start with an electrical potential difference across this membrane of negative 70 millivolts. Now, what does that mean? Well, the electrical potential difference across a cell membrane actually means the electrical potential difference from the extracellular compartment to the intracellular compartment. So it means if you have a little man standing in the extracellular compartment who measures electrical potential, and he then moves into the intracellular compartment and then measures electrical potential again, he'll get 2 values. He'll get the electrical potential of the intracellular compartment here, and he'll also get the electrical potential of the extracellular compartment. Now we can ask, how much does it change if you move from extracellular to intracellular? How much does that number on your little machine change? Well, basically that will be the electrical potential of the intracellular compartment subtract the electrical potential of the extracellular compartment. That is what we mean by the membrane potential, or the electrical potential difference, or the voltage across the membrane. It's the difference in electrical potential between the extracellular compartment and the intracellular compartment. And it has to be directed. So when we talk about normal membrane potential, we mean from extracellular to intracellular, not from intracellular to extracellular. That would just be the negative of this. Okay, so it's usually... Well, that's the thing about these cells. They don't have a resting membrane potential. Instead, they just continuously go through a cycle. But at the moment, our starting point is that it's going to be negative 70 millivolts, which means that the intracellular compartment has a lower electrical potential than the extracellular compartment, and it's lower by 70 millivolts. Okay, right, now at this electrical potential difference across the membrane, these channels are opened. They're activated to open. So the HCN channel opens, which is odd. Mainly, channels open when the membrane becomes depolarized, not hyperpolarized. So this is what makes these channels odd, and this is why sinoatrial node cells fire action potential spontaneously, because of the presence of these HCN channels, which are mainly HCN4 homotetramers. Okay, right, so what is the HCN4 channel permeable to? Well, it's permeable to two ions. It's permeable both to sodium ions and to potassium ions. So, I'm going to give you the concentrations of sodium and potassium on either side of this membrane, and in the break, you can try and decide what is actually going to happen. So, the concentration of sodium in the extracellular fluid is approximately 145 millimolar, okay? The concentration of sodium in the intracellular fluid is approximately 12 millimolar. Okay, so a concentration gradient of around 12-fold, favouring the movement of calcium into the cell. Sodium into the cell. So this is the sodium ion. Okay, and then also the potassium concentrations are 4 millimolar extracellularly and 155 millimolar intracellularly. So these are the concentrations, and remember the electrical potential difference across this membrane. The intracellular compartment is lower in electrical potential by 70 millivolts than the extracellular compartment. Okay, I'll see you in the next video.